Much. Thanks for the kind invitation for Katia, Anna, Martin, and Minimo, aka Sandip. I'm not going to show any embarrassing pictures. So anyway, I'm going to talk to you about some of our work on crack channels, and what I'm going to talk about, it's mostly native crack channels. So if you see an overexpression, that will be within the context of understanding the native channel. And so perhaps this has already been shown ad nauseum. The two molecules involved in this pathway, STIM in its inactive state, which in response to store depletion loses calcium from the EF hand, rearranges, gains this extended conformation, and then it moves to ER plasma membrane junctions, where it traps and activates hexameric ORI channels. And so this calcium entry pathway, ubiquitous, perhaps in addition to refilling the stores, its role, its signaling, and most notably through calcineurin and downstream NFAT transcription factors. So for some time now, we were interested in actually vascular smooth muscle remodeling. And some of you might know that unlike cardiomyocytes and skeletal muscle, which are terminally differentiated, a vascular smooth muscle maintain this plastic phenotype. They maintain the ability to de-differentiate and to go back to this more kind of stem-like, so to speak, phenotype. And that's because it's perhaps an evolutionary acquired kind of mechanism for wound repair and healing, for maybe adaptation to stress, to exercise during pregnancy, etc. But this differentiation can also be a hallmark of disease. And one of the models that we've studied in the past is this restenosis model, which normally you go in and you apply this balloon angioplasty in the coronary artery to restore blood flow for people that are having a clogged artery. But one of the consequences of this mechanical stimulation is this neointima formation. So you can look at the normal vessel here. You have the intima, which is endothelial cells in direct contact with the blood. You have the media, which contains the smooth muscle cells. Those are the ones that regulate vascular tone. And you have the adventitia, which contains a collection of cell types, including fibroblasts and connective tissues. So at day 7 after injury, you can already see the formation of this neointima. And the peak of this model is right here. All this is basically occluding the lumen. And you can even see that those cells here in the media are kind of mostly hypertrophic and like actually undergoing a growth phase. And this is typical. I mean, this is a nice model, but this smooth muscle differentiation is also a hallmark of things like atherosclerosis and hypertension. And so quite some time ago, we've shown actually that in spontaneously hypertensive rats, that the stim is upregulated and also the ORIs are upregulated. And then in this, I'm skipping a lot of data. I made a promise that I'm not going to show any bar graphs, just to kind of, keep it simple. So if you look at the iCrack or the current, and you can go to these cells here, and you can really remove these cells, and then dissociate them enzymatically and do patch clamp. You can see that the cells from the media as well as the intima have this typical iCrack with a positive reversal potential, suggesting that this pathway, actually, you cannot find this crack currents in the quiescent smooth muscle. There is some level of stim expression, but there is essentially no crack to be measured. So it shows up here in the media and in the neointima, suggesting that it's an early kind of mechanism to drive this smooth muscle de-differentiation, which is characterized by proliferation and migration. And basically, I'm skipping a lot of work here, but this is just kind of the last slide of this paper where we showed that through mechanisms that involve in CAM kinase 2, CRAB, and NFAT, that's how actually the crack pathway drives this smooth muscle de-differentiation. Here in the rat, if you deliver shRNA against luciferase as a control, you could see this neointima. But if you deliver shRNA, this is with the lentiviral vectors, transduced for just 20 minutes within a section of the left carotid artery, you could see you can reduce this neointima quite significantly. And so with that, actually, we moved on. And one of the things it was quite an opportunistic move, so to speak, which is trying to look if this is kind of holes in things like airway smooth muscle. And we were having our eye on asthma in particular because of the role of airway smooth muscle remodeling during asthma. So we made the knockout mouse and the smooth muscle specific knockout mouse using this MYH11 cray. Actually, the previous smooth muscle 22 alpha is really not a good one because it has a lot of extravascular expression, including the kidney and the liver and the pancreas. So that's a little bit. So the angioplasty model in the mice is pretty much impossible, by the way. So just so you know. So this is just when you isolate smooth muscle specifically from the airways of this mice, there's not much stream there. It's reduced by 90% or more. And I hope this will work. And if it doesn't work, that means that it's this is, I just show it, but what should I do? Okay, let me get my glasses. Okay, so while I am trying to figure out show pointer option, yeah, can you thank you. So this is basically a live section of thank you of airways, and where the cells smooth muscle are expressing the GCAM6, and then those are challenged with methacholine. And then that's just showing an example of, you could see, I hope you could see the airways kind of contracting. You could see those oscillations. Some of them are raised oscillations, and some of them are just quite like going back to baseline. And that's basically typical what you see in these airways. And so when you do the same experiment, I'm actually again skipping the movies and stuff for lack of time. You could see those oscillations that let me go back to the pointer option again. There you go. These are some of those oscillations in the control mice. You can see they're over a plateau here and stuff. And this is the oscillations that you see in the smooth muscle specific STIM1 knockout. You have less oscillations. Some of them have this tiny little amplitude. Sometimes you get this big dump at the beginning, but then nothing after. But they don't look anything quite like this. This is analyzed like hundreds and hundreds of actually regions. And again, I'm not showing any bar graphs. You can go to the paper and look at the quantification of all this data. One thing that was really very interesting by looking at all in the bronchoalveolar lavage of all these animals, none of the leukocyte, number of leukocytes are changed. Everything looks the same. But here what I'm showing you, I lied actually, this is a bar graph here, but anyway, hopefully this is the only one. It's an IL-6, and this is IL-6 in the bronchoalveolar lavage. You can see that's mostly in actually the house dust mites challenged, which is the model of asthma, Cree mice, which are the control, but not much in the STIM1 smooth muscle knockout. So this really suggests that actually the smooth muscle in these situations behave really like an immune effector. And in the paper, you can go and look at, we did some kind of RNA sequencing and connected some mechanistic biochemistry, connecting NFAT4 to actually IL-6 specifically in smooth muscle. So this IL-6 is coming specifically from the smooth muscle. And that's a respiratory mechanics data looking basically at the increasing doses, the airways of live mice challenged by increasing doses of methacholine. And you could look at the two control here. Those are two types of controls. Those are like, because this is a tamoxifen induced, these are just the floxed mice. And those are the Cree mice, and so the flox mice are actually not challenged with anything. And that's where the STIM1 knockout, smooth muscle specific knockout mouse fall. So there is a significant reduction in this airway resistance in response to increasing methacholine challenge. And so you could conclude from this that maybe, perhaps, that targeting crack channels for inhibition during asthma could have a double beneficial effect, because we know now that there are some drugs in phase 3 clinical trials, against specifically ORI1 for rheumatoid arthritis and things like that. and the cytokine storm, I believe, for the case of COVID. And so you could imagine a scenario where if there is an aerosolized drug given to asthmatic patients that will work both on the smooth muscle components and on the immune components of the disease. So that's the idea anyway, that's the theory anyway. But I think that this is probably a very simplistic view, and the reason is because I think I've been alluding to this before in some of my talks, is that there is 3 ORIs in mammals, ORI1, ORI2, and ORI3, and there are 2 stims, as Jen told you this morning. And so this makes things a little bit complicated, because when we looked at the HEC293 system, where we did single, double, triple, quadruple, quintuple knockouts, sure, we could confirm that ORI1 is the major channel in response to store depletion. Look at the red trace, it's the calcium entry is almost completely gone. But I'm oversimplifying the data here. If you do ORI2 or ORI3, or in this case here, the double knockouts, this signal actually increases. And so the conclusion from this is that ORI2 and ORI3 are negative regulators of ORI1 channels. And this is one of the evidences that you could look here, and you could take wild type cells and challenge them with increasing doses of carbachol, and then they start kind of oscillating, but then they plateau at higher doses. This is the wild type cells. But then if you take the ORI1 knockout, they just continue to oscillate with the same frequency, and they will never, ever show you a raised plateau because they don't have ORI1. However, when you take the double knockout, well, 2 things happen. They start responding earlier, and they start plateauing earlier. Why? Because they lost now the brakes, they've lost the negative regulators. And so we think that the native channel is oversimplistic to think it's just an ORI1. It might be in some cases, but in other cases, it might be a combination of all the ORI1 or ORI2 or ORI1 and ORI3, or maybe all the 3. And how would this work? Well, if you, this is an overexpression experiment where we kind of measuring YFP, ORI1, ORI2, ORI3, co-expressed with non-limited amount of stim, and doing this calcium-dependent inactivation. And I hope you could see that that ORI3 inactivates more than ORI2, which inactivates more than ORI1. So we think this is a potential mechanism how an incorporation within the hexamer of one of these subunits will serve as a brake, as a negative regulators. And we've done, not only as others, done experiments with concatemers or with co-expression, and the CDI is a hybrid of these two channels when they're expressed together. So looking down at NFAT, in this case, again, this is the native here. And if you look at HEC, just focus on this is the translocation of NFAT1 in response to carbachol. This is the translocation of NFAT4. If you look at HEC, you can see that's the black trace. If you don't have ORI1, nothing much really happens. But if now you lose ORI2 and ORI3, now NFAT translocation is accelerated and much more faster. And now what happens is that the translocation of NFAT4 and NFAT1, which were supposed to be basically temporally distant, because look at the scale, this is 14 minutes, this is half hour. Now they look almost the same, so it's almost like because of lacking these negative regulators, there's no distinction between the concentration range with NFAT1 versus NFAT4 should move to the nucleus. So it's almost the cell lost its ability to distinguish between these 2 transcription factors because you're missing that negative regulator. And so we kind of pontificated a little bit, and there's about this in saying that in vertebrates like Drosophila, you have 1 stim, 1 ORI. It's probably kind of like an on-off switch, while in mammals, there are certainly evidence that ORI3, ORI2, and ORI1 can exist as homohexamers, but then there is possibility that those will heteromultimerize. And that's the same true perhaps for stim. And this serves as a fine-tuning, or I guess that's the hand waving that we all use to match the complexity of calcium signaling in mammalian cells. And now you would say, well, this is HEC293 cells, and who cares about HEC293 cells? And I would agree. So in the last 3 minutes, I will show you some data on actually primary B cells. And this is primary B cells where we use the MB1 to generate single ORI3, single ORI1 knockout, B cell specific, and double ORI1, ORI3 B cell specific. And this is a calcium trace where here with ORI3, it doesn't look much like, it doesn't look like it's inhibited or anything like that. So the only hint for the people here, the 2-APB aficionados, they will notice that this inhibition by 2-APB is a lot faster in this case when you remove ORI3. That's the only difference we could see. But anyway, this is the knockout of ORI1 alone. When we do the double knockouts, they're additive. So although this data does not really conclusively tell you that ORI1 and ORI3 are probably heteromultimerizing, but at least it tells you that both of them are involved. And I think the next part of the data is that this is looking at NFAT1 translocation. Just focus on this. This is dephosphorylation of NFAT. This is the naive T cells taken out of the animals. You can see this is a lot of the NFAT dephosphorylation is kind of inhibited compared to the control where dephosphorylation is complete. But if you look in the cells that are treated for 48 hours with IgM and CD40, the co-stimulation, and actually the mature cells or the activated cells, a lot of it is inhibited. So this NFAT signal is really required for actually the cells for clonal expansion, proliferation, and so on. And going back to this metabolism and this kind of idea, you could see here just in this oxygen consumption rate using this seahorse assay that most of you must know. You can just see here the red trace and the teal trace, which is the ORI1 knockout and the double knockouts, and to some extent the blue trace. The basal respiration is reduced, and then the maximal respiration here is marginally reduced for ORI3, not significant, but definitely for ORI1, but additive for ORI1 and ORI3. When you do this chart, this map of OCAR versus ACAR, you could see that the teal trace sits right here, which is more so than the red trace. So these cells are really quiescent. So that's why they cannot proliferate very well. They cannot secrete antibody and do clonal expansion and things like that. And so this is a complicated slide, but you can this is just metabolite looking at just nucleotide, amino acid, small molecules, precursors, and things like that. And the controls are here, and the double knockouts are here. You just compare this green square to this green square, that's stimulation with IgM. The red is stimulation with IgM plus CD40. You can clearly see here that it's not as robust as the case here. And then the control, we're using FK506 as a calcineurin inhibitor and the GSK as a SOC inhibitor. So clearly there is a metabolic effect. And this is a theme now, this crack channel connecting to metabolism via NFAT and via IRF4, via MYC, and that sort of things. This is a theme that is common. I mean, the work from Stefan Fiske on T cells, Murali Prakriya on astrocytes, ours on smooth muscle and B cells. So this is I think it's how this pathway controls kind of growth. And so then go back to my conclusion is that my final conclusion is that maybe this is kind of a blunt approach until we understand more of the channel composition. So we know actually what cell type expresses what specific channels. Well, unless we get lucky, like with CAV 1.2 and hypertension, because if somebody told you at the beginning that you could use CAV 1.2 blockers, a channel that is required for beat to beat of the heart for treatment of hypertension, that you would say that's the stupidest idea I've ever heard. And yet it works, and we know it works because the binding of these channels to CAV 1.2 is voltage dependent, and that's smooth muscle is tonically contracted, and that's why they work. In addition, being a splice variant, so in essence, maybe it will work. We'll see, I guess, what's happening with these ORI1 blockers. But then, as more blockers that take into consideration ORI2, ORI3, and maybe how much ORI2 and how much ORI3 are in the complex. And what we're trying to do now is use to some knock-ins under native conditions to get down to the stoichiometry, because no amount of cryo-EM or X-ray crystallography is going to answer these questions under native systems. And with that, I think I cited the people as I go, the NIH for funding and the current lab members, and